My name is Emily Beardon and I'm a plant health scientist at the Animal and Plant Health Agency. I did my PhD at the University of Sheffield in Julie Scholes' group, and my PhD was on Striga. Striga is a plant, it's a weed, but it's also a parasite. So it starts its life cycle underground as a tiny seed that's about the size of a little bit of dust. So it's impossible to see really with the naked eye in the soil. The first thing farmers know is that their crops aren't growing very well, stunted growth. And then when it gets big enough, the plant will emerge from the ground and flower and produce lots of seeds that will just blow around in the wind. Striga attaches to the roots of the crop plants, and it actually forms direct xylem connections with the roots. So it will drain the nutrients and the water from the plant and stunt its growth. And it can actually cause devastating crop yields. So it's a real problem mostly for subsistence farmers who can't afford expensive fertilisers. My PhD was actually looking at the genetics of resistance to Striga. There are some varieties of rice, I was working on rice, that actually show quite good resistance to the parasite. And my PhD was specifically trying to identify the genes that were involved in that resistance. The ideal scenario would be that we could breed multiple resistance genes into a single cultivar, and that would then provide broad spectrum resistance to the parasite and help control the parasite and improve yields for farmers. So finishing my PhD, I then moved to Norwich to work at the John Innes Centre, and that was to do a postdoc on ash dieback disease, which is a fungal disease that affects ash trees. The disease is a fungal disease. It originates from Asia, and there it lives fairly harmlessly with native ash. But in Europe, it's actually quite harmful to our trees. Our trees here didn't evolve with that parasite. There are only a small number, maybe 5 to 10% of trees that have some form of resistance. Most trees get infected and will eventually die. We don't fully know how it arrived in Europe. We do know that it was identified in Eastern Europe, I think in the early 90s. It was identified in the UK in 2012. And surveys were carried out, and from that we found out that it had actually spread into the wider environment. We think now that it was imported from bringing ash trees in from Europe, but also we know, or we suspect, that spores were blown across the channel and in fact infected trees in the southeastern part of the country. Spores will land on the leaf surface and then will grow through the xylem vessels and will block those vessels, so that will cause wilting of the leaves, browning and wilting, and then it will grow through the leaf stalk and into the stems and eventually the trunk, and it will block those vessels and that will cause the branch and eventually the tree to die. It's pretty serious. Ash dieback's now pretty much all over the country. You can find it wherever you can find ash. If you see an ash tree, chances are it will probably have some signs of infection, and many trees have now died. But there are also some trees that are showing some form of resistance, so there is hope out there still. I was investigating a possible trade-off between ash dieback resistance and resistance to insect pests. So the worry was that those trees that survive ash dieback may be more susceptible to insect pests, and I was investigating that trade-off. I also was looking at the transcriptomics in response to infection. So I infected a variety of trees that had different forms of resistance, so some susceptible trees and some resistant trees, and I looked at how they responded to the infection at the genetic level. Transcriptomics is looking at a kind of snapshot of what is actually happening in the cell at that time. So what genes are being expressed, and we can sequence those genes by sequencing the messenger RNA, and then we know what genes are turned on. So back in my first year of undergrad, I came to the Gatsby Plant Summer School, and I really enjoyed that, particularly the plant pathology practical actually, and after that I ended up picking a lot more of the plant science modules for my degree, and that's the kind of the direction my degree took. I did a master's in plant pathology, and I really enjoyed that. And since then, I've always been interested in plant interactions with other organisms, whether that's diseases, whether that's pests, whether that's parasites, and that's what I continue to do. So APHA, or the Animal and Plant Health Agency, is the agency that's responsible for safeguarding the UK against pests and diseases. My background is plant science, and actually I work in the international development team, which is a bit different, and I work on a project based in South Africa. The project is an official development assistance project, and it's partnering with South Africa because South Africa already has the universities and their expertise to help us with the research. And the aim is to create food systems that are climate resilient, biodiverse and sustainable. What I really love about the project is seeing the bigger picture. So it's really inspiring to work on a project that is really trying to see food systems as being dependent on the environment and also affecting the environment. It's all about sustainability in the food system.